So we continue with the series on fetal membranes. We had talked about the fate of the yolk sac. Sorry, this is still Dr. Ominde. So yeah, so what is the fate of the yolk sac? We have what we call the Meckel's diverticulum. When it persists, it forms a Meckel's diverticulum. So you can have a fistula, and I said a fistula is a communication between two surfaces that are lined by epithelium. So in this case, you have your diverticulum connecting to the skin, and there is a connection from the GIT to the skin, and that's a fistula. Then you can form a Meckel's cyst, okay, just a fluid-filled cavity, or you can form a Meckel's sinus that it's just with one, it doesn't open on both sides, okay. So this is your Meckel's diverticulum. So this is normal bowel of your GIT, but there is that protrusion that becomes your Meckel's diverticulum. Ideally, the yolk sac should obliterate, but if it persists, you get your Meckel's diverticulum, and it's usually attached onto the anterior abdominal wall. So this is how it looks like in the abdomen. This is your anterior abdominal wall. That's the umbilicus. So next we go to chorion. So how does the chorion form? The chorion is usually from somatic intraembryonic mesoderm, and it has two parts, the cytotrophoblast and syncytiotrophoblast. So this is your chorion here. So you have the cytotrophoblast, and syncytiotrophoblast is the one that invades the deciduum during implantation. So what are the functions of chorion? It plays a role in hemopoiesis and also helps to protect the fetus by protecting it from toxic substances in the maternal circulation from entering into the fetus. So this is your chorion, and remember we talked of chorionic villi in the previous lectures, where you have primary villi, secondary villi have mesoderm, and then when they form blood islands, they become tertiary villi. So these are the villi with the intervilli species. Then we go to the allantois. So allantois comes from the endoderm. Okay, it comes from the endoderm, and it's a protrusion from the yolk sac. So it's a tubular endodermal outgrowth from the dorsal end of the yolk sac. Then usually it will extend into the primary mesoderm of the connecting stalk. So an endodermal outgrowth from dorsal end of the yolk sac extending into primary mesoderm of the connecting stalk, and it's associated with formation of cloacal membrane towards the anal opening. So this is your cloacal plate here, and this is your allantois, dorsal protrusion, endodermal protrusion from the yolk sac. You can see all this is yolk sac, so when it extends. So what are the functions of the allantois? It helps with transfer of nutrients from the mother to the fetal circulation, and then some germ cells can originate from allantois, but germ cells are mainly from the yolk sac, and then allantois could be part of what forms the umbilical vessels. So the allantois vascularizes the chorion and its villi with the allantoic or umbilical vessels. What's the fate of the allantois? It forms the urachus. The urachus is just a distal fibrous part, and the proximal part of the allantois is usually incorporated into the bladder. So the allantois has a proximal part and a distal part. The proximal part is incorporated into the bladder. The distal part becomes fibrotic and forms the urachus. Then the allantois also forms a median umbilical ligament. So this fibrotic material at the midline forms the median umbilical ligament. So an abnormality comes in where this urachus, instead of undergoing fibrosis, it remains open, and then it causes a communication between the urinary bladder and the skin, giving you a fistula, communication between two epithelial lined surfaces. So you get a urachal fistula. So this is what we mean. This is your urinary bladder. So allantois is here. Ideally, it's supposed to obliterate. So the proximal part is incorporated in the bladder. The distal is supposed to obliterate. But if it fails to obliterate, it remains patent, and you can see it's communicating the bladder with the anterior abdominal wall at the umbilicus. So this patient will be leaking urine through the umbilicus, urachal fistula. So ideally, proximal part incorporated in the bladder, distal part forms the urachus, and with the peritoneal fold around it, this urachus forms the median umbilical fold or median umbilical ligament. This is it. This is the median umbilical fold. Okay, this is your bladder. Then we go to the umbilical cord. The normal umbilical cord is usually approximately 50 centimeters. What does it contain? It contains connective tissue jelly. We call this jelly the Wharton's jelly. Umbilical cord also contains one umbilical vein and two umbilical arteries, or a pair of umbilical arteries. And it also contains the degenerated yolk sac and allantois. So those are the contents of the umbilical cord. Wharton's jelly, one umbilical vein and two umbilical arteries, and degenerated yolk sac and allantois. Umbilical cord is covered with amniotic membrane, and usually it helps to connect the fetus with the placenta. So abnormalities of the umbilical cord, it can be too long or too short. When it's less than 35 is too short. When it's more than 80 centimeters is too long. So in a full-term fetus, usually the cord will measure 1 to 2 centimeters in diameter and 30 to 90 centimeters in length, with an average of around 50 to 55. Then this cord is supposed to be attached at the center of the placenta, though you can have insertions at the marginal part, or you can have the velamentous insertion. So you could be asked, what are the variations of umbilical cord? So this is your placenta, this is umbilical cord. So you can see this one, this is your placenta, this is the umbilical cord. Okay, so it inserts onto the fetal surface. This is the fetal surface of the placenta. So this is what you call the battledore placenta. The umbilicus, it's supposed to be at the center. You see this one is at the center, but this one is at the margin of the placenta. So that's battledore, marginal. But you can also have velamentous placenta. What do we mean? You can see this placenta, the umbilical vessels touch the placenta before they branch. Even in this one, they've at the surface, they touch the surface of placenta before the branching. But in velamentous, the cord is here, it gives branches, so it's the branches that actually get to the placenta. So this is your umbilical cord, this one here, connecting the fetus to the fetal part of the placenta. So the placenta at birth weighs 500 grams, and it has 15 to 25, it measures 15 to 25 centimeters in diameter, and it's 3 centimeters thick. Usually it contains anchoring villi, and the maternal surface, it has a fetal surface and maternal surface. The maternal surface has 15 to 20 projections, which you call cotyledons. So this is your umbilical cord, this is the fetal surface is smooth, and this is the maternal surface. You can see these are the cotyledons. This is the placenta here. Fetal surface is smooth, maternal surface has the cotyledons. So, and these are the anchoring villi that are in the placenta. So usually it's discoid in shape, okay, and we call it a choriodeciduate organ, connecting the chorion to the decidual basalis of the endometrium. So it connects the fetus with the uterine wall of the mother. Therefore, you have maternal and fetal tissues coming in direct contact without rejection, because you have immunological acceptance of the fetal graft by the mother. So immunologically, there is that acceptance to allow the two of them to contact without any immunologic rejection. So the fetal surface, we've said, is usually smooth. It will be covered by amnion and has an attachment for umbilical cord. While the maternal surface, we've said, is rough and irregular because of the 15 to 20 polygonal cotyledons on the maternal surface. So the placenta is formed by two portions: the chorionic frondosum from the fetus and the decidual basalis from the endometrium. It's 15 to 20 in diameter, and it's formed sort of by a sandwich. The fetal part, which is the chorionic plate, and the basal part from the mother, but in between you have the chorionic villi. So the basal plate is formed by the decidua and the cytotrophoblastic shell. So the placenta maternal portion has all the placental septa dividing the placenta into 15 to 20 lobes, and these are the cotyledons, which are incomplete lobes. So again, This is the image of a placenta. You can see the cord coming to the placenta, and you can appreciate you have this decidual basalis. So this is the maternal, and this is the fetal surface that is smooth, and you can see the septa dividing the placenta into 15 to 20 cotyledons. So the structure, placenta membrane is just a structure between the blood of the mother and the fetus, and this is basically formed by sensitive trophoblastic cells invading into the decidua. So you still have some part of the cytotrophoblast lying on basal lamina, and then connective tissue as well as the endothelium of the blood vessels with their basal lamina. So these are what forms the placenta. Syncytial and cytotrophoblasts on the basal lamina, connective tissue, and endothelial cells lining the blood vessels lying on their basement membrane. So this is your fetal membrane. You can see syncytial trophoblast here, the cytotrophoblast here. They're lying on a basal lamina. Then there's connective tissue separating them from the blood vessels. And these blood vessels are lined by endothelium lying on their basement membrane. So this is the maternal blood bringing nutrients to the fetal circulation, and you have products of waste of metabolism from the fetus into the maternal circulation. So the placenta will help to exchange or carry out transport of oxygen, nutrients, carbon dioxide, waste, electrolytes, and antibodies. It produces hormones such as human chorionic gonadotropin hormone, human placental lactogen, progesterones, and estrogen that support the developing fetus. Then the placenta carries out metabolism in early pregnancy. It's able to synthesize glycogen, cholesterol, fatty acids, and this produce a source of nutrients and energy for the embryo. The placenta is also a defense barrier, okay, to protect from toxic substances in the maternal blood from entering into the fetus. So this is how exchange oxygen and nutrients from the mother, and then salts and water could be both ways, carbon dioxide and metabolic waste from the fetus to the mother. Then the placenta acts as a barrier. So if you have microorganisms in the maternal blood, there is no way they will get into the fetal circulation because the placenta acts as a barrier. So some drugs also in the maternal blood will not be able to access the placenta to go and cause an effect onto the fetus. So next we discuss twinning and multiple births. Of course, you know we have two types of twins. We have monozygotic twins and dizygotic twins, and then multiple births can be monozygotic multiple births, polyzygotic, or mixed multiple births. So what happens in dizygotic twins? Remember, after fertilization, there's cleavage, then you form a blastocyst. So from the blastocyst, you can form the two amnions, amniotic membrane, two chorionic membrane or cavities, and two placentas. So each of the blastocysts each of the embryo will now have an amnion, a chorion, and a placenta. That gives you dizygotic twin. Okay, but monozygotic twin, it means there was a separation at the blastomere, or you can separate at the cell mass, or you can separate at the time formation of the primitive streak. So when the separation occurs at the blastomeres, you get what we've seen in dizygotic twins. You get two amnions, two chorions, and two placentas. If the separation occurs at the inner cell mass, you get two amnions, but there will be one chorion and one placenta. But if there's a separation at the level of the primitive streak, you get all the two twins sharing an amnion, sharing a chorion, and sharing the placenta. Okay, so I hope that is clear, because if the separation of the embryo occurs at the blastocyst stage, each of the twin will have their own amnion, chorion, and placenta. So there will be two. If there will be separation at inner cell mass stage, remember the blastocyst will develop spaces which will gather fluid and separate the cells. So you have inner cell mass and the blastocele. So the inner cell mass, if you separate at that stage, you'll get two amniotic cavities, but these two twins will be sharing the chorion and the placenta. Then later at gastrulation level, this is way later. This is the early stage of separation. You get two. Then later on, they only share the amnion, and everybody gets different amnion, but they share chorion and placenta. But if you do later separation, each twin will have the two twins